I'd like to welcome you to Genomics England Research Seminar Series. We're going to have two talks today from Serena Nik-Zainal and Alistair Pagnamenta and Marcello. And just before we get started, just make a few announcements about these seminars. Firstly, so everyone notes, and I'll just repeat this a bit more as more people have joined, everything is being recorded. So if you want to ask a question, put questions into the Q&A rather than put your hand up at the end of the talks. So we're going to have a Q&A after each of the two talks. Can we move to the next slide? So just to remind everyone that these talks we have today, they're all based on use of Genomics England 100,000 genomes data. If you haven't got access to this data, doesn't matter whether you're an academic or industry, you can apply for access to join one of the structures, the domains around different disease areas. You just have to go through a process of signing up and getting your institution to sign up if it's a healthcare institution, and you can get access to this closed environment and do analysis of data there. At the moment, that's free in terms of access. If you're an academic, researchers have to pay for access. But again, it's the same principle for data analysis within a closed environment with all the genome data and a large amount of clinical data, all under the ethics of the 100,000 genomes data, Genomes Project. Next slide. If you want to check this out, then one way of doing that anyone can use is to go to the research portal. You can see all the projects that have been registered there. Lay summaries are available. If you're actually in the project, you can log in and see the individuals and more details about each project. If you want to follow what's coming out in terms of research, there's a specific Twitter account for this, where we announce everything that's coming out in terms of research publications. And if you want to know more about what Genomics England is doing and the engagement around genomics in general, there's a new podcast hosted by our CEO, Chris Wigley, with a range of guests from major scientists in the area of trying to bring healthcare, genomics into healthcare, to patient participants and those who are dealing with issues around ethics. So there's a range of talks for you to go and listen to. And if you've got any suggestions for talks you would like to see at this seminar program, then there's an email address down there. In terms of the seminar program, we're having it regularly, the last Tuesday of every month at 2 o'clock. So the next one's on the 27th of April. Next slide. So I think we're going to start pretty much now, where participants are up to 220. So I think that's probably good to get going. So the first talk is by Serena, and this is the breast cancer domain of Genomics England. And she's going to talk about new insights of mutational signatures from the 100,000 Genomes Project data. So Serena is a CRUK advanced clinical scientist and honorary consultant at the Clinical Genetics in Cambridge. And she's known for having really introduced this idea of mutational signatures present from analysis of whole genome sequence data. So Serena, over to you.
Thank you very much for the introduction, Tim, and for the opportunity to present today. So hopefully you can see my slides in full mode rather than in presenter mode. And so I'm going to focus today on recent updates on this concept of mutational signatures, actually on behalf of the pan-cancer domain rather than the breast domain. So I'm part of two GSIPs. But really, in the interest of time, I'll focus on some of the aspects that have required the use of cancer data from the 100,000 Genomes Project. So the good thing about talking to all of you is I don't have to try to convince you that whole genome sequencing is the test to do. And the fabulous thing about whole genome sequencing is that there's so much data that it allows us to see far more in a cancer genome than that handful of 1 to 5 driver mutations that have commonly been sort of the focus in cancer research. So human cancer acquires not just those drivers, it acquires thousands and thousands of passenger mutations. And they arise because of the various mutagenic processes that are going awry. So some mutagenic processes may be perfectly normal and tolerated in cells perfectly okay, like replication errors. Some are due to environmental exposures like tobacco smoke or UV light. Some mutagenic processes, however, may be a little less okay. They may indicate a defective DNA repair pathway, for example. But each of these mutational processes leaves a characteristic mark, an imprint on the genome that we call a mutational signature. When you sequence a cancer genome, what you get is a final portrait. It's a composite of all of it added together. But when you have many genome sequences of the same tumor type, it is possible to decompose what looks like a random mess of mutations. It's possible to actually identify the individual patterns, the individual signatures, as well as to quantify the amount of them. So basically find out the size of these arrows. And we've shown this in the past when I was at the Sanger Institute. So historically, during my PhD, we presented signatures like this. These are base substitutions for the simple reason that base substitutions were the easiest class of mutation to call. So we present substitutions as 96 channels because for every base, we take the neighbors into account. But this is just to show you that the signatures are really defined or have characteristics that are recognizable. So in this case, signature 1 has tall peaks, red bars here at CPGs because this is the signature of deamination of methylcytosine. It's perfectly common and normal and happening in all your normal cells right now. And there are many other intrinsic or endogenous mutational processes happening in your cells, but there are also exogenous or environmental agents that can leave characteristic imprints on the genome, actually well before whole genome sequencing could show it in spades. People were already doing these sorts of experiments in a petri dish way back in the 1990s to show that there are characteristic C2T patterns indicative of UV light that you can see in skin cancers. This is the signature of tobacco smoke that you can see in lung cancers. And this is a classic signature of something called aristolochic acid that you can find in liver and kidney cancers. So I will say it's early days. The number of mutational signatures isn't static. There are signatures of other classes, double subs, indels, rearrangements. And the signatures are derived from noisy cancer data sets, right? Patients are not controlled in any way. So we do need some validation from additional data sets like Genomics England, and we do need validation from experimental work as well, which brings me to my next section on experimental validation. So we've been systematically performing experiments to generate mutational signatures under experimentally controlled conditions. Can we recapitulate what we see in cancers? Can we find new ones by taking a systematic approach? And the short answer is yes, we can. We've published a compendium of mutational signatures of all the environmental mutagens that are known or suspected to be implicated in cancer in this paper in 2019. I won't go into it today in the interest of time, but I will say that you can access the signatures, the patterns themselves. You can browse the experimental data. You can download the data directly from this website here, which has been set up by these guys in my team. Today, I'd like to focus on this half of the experiments because of how we leaned on Genomics England data to clinch clinical utility of this work. And this was done in collaboration with Bill Scarnes and Joe Jurikny, spearheaded by Shuqing Zhu in my lab, super talented analyst. And so this is a paper that's in press. We knocked out using semi-high throughput methods, that's CRISPR-Cas9, a whole bunch of DNA repair and replication genes. And crucially, we didn't treat the cells with anything else, no DNA damaging agents, no alkylating agents or platinum or anything like that. We just knocked out the gene. Did we get a signature? Because if you do, then that gene is really important at maintaining the genome, at sanitizing the genome from endogenous DNA damage. In the interest of time, I'll focus on this family of genes, the mismatch repair pathway, a highly conserved repair pathway that plays a key role in reducing the error rates at replication, amongst other things. When the MMR pathway is compromised, tumors have historically been described as having microsatellite instability, lots of little indels, and a high tumor mutational burden, or TMB, MSI and TMB. But in fact, when you knock out mismatch repair genes, you see much more granularity through whole genome sequencing experiments than just MSI or TMB. There are quantitative differences in the substitution and indel burdens between the different genes. There are also interesting qualitative observations to make between them. So you can see the knockouts of MSH2, MSH6, and MLH1 produce nearly identical mutational signatures, and yet PMS2 looks really quite different. So we have differences in substitution profiles and also indel profiles as well. So just to orientate you, these are T deletions. You get more of them as your polynucleotide repeat tract. That's your TTTTT, gets longer and longer, which kind of makes sense. MSH2, MSH6, and MLH1 look very similar to each other, just like the subs, but PMS2 looks rather different with about equal representation of T insertions as well as T deletions. So there's this sort of gene specificity that you can see with mismatch repair knockouts. And we validated this gene specificity in other models. We derived iPS cells from patients who had constitutional mismatch repair deficiency. So these are usually children with a syndrome as a result of having inherited biallelic mutations. So they've got basically Lynch syndrome genes, but they've got two of them. They get cafe-au-lait patches, and they often succumb to pediatric malignancy. So this is a severe phenotype. We made iPS cells from them, and we just grew out the cells. We didn't do anything else. And the question is, do we see mutational signatures in their normal stem cells? And do they recapitulate what we did when we knocked out the genes? So here are the knockout genes, and here are 4 different sets of iPS cells, 2 from MSH6 mutated patients and 2 from PMS2 patients. So there's 4 patients altogether. And you can see a very high concordance between the signatures in the patients and the signatures in the knockouts for the MSH6 and PMS2 in this experiment. So we validated it in another experimental model, and we find this very strong concordance. That's great. What do we do with this information? As my mother once said when I finished my PhD, you've got a PhD in pretty patterns. How does it benefit mankind? So we do work quite hard to try to create clinical algorithms in the team, and we developed a logistic regression classifier called MMR-Detect, using those mutational signatures that we had identified through the experimental studies. But critically, we were able to use the 336 colorectal cancers that were present in Genomics England that also had clinical pathology data. So they had immunohistochemistry of the mismatch repair proteins, that's MSH2, MSH6, MLH1, PMS2. We all know in the clinical domain that this is something that is kind of used in the NHS, but it was really valuable to have access to that sort of data and not just have the genomic data. So we're able to randomly assign the patients then into a training set and a test set. We did tenfold nested cross-validation and developed the weights for the algorithm, which we called MMR-Detect. Now, as a comparator, we applied another widely used classifier on NGS data called MSI-Seq, where it looks just for indels, indels at polynucleotide repeat tracts, so that we can, do a sort of systematic comparison of immunohistochemistry, MMR-Detect, and MSI-Seq. And here's the results. We know that IHC, immunohistochemistry, finds 79 cases of mismatch repair deficient patients. We found 77, and MSI-Seq found 79. What's the overlap? So the overlap's actually very good. 75 of 336 were concordantly defined as MMR deficient by all 3 methods. Now, there were 4 cases that were found to be MMR deficient by immunohistochemistry, but not by the other 2 algorithms. And in fact, we did not find a genetic defect. We couldn't find a mutation in any of the mismatch repair genes. Now, for these 2 cases here, which were shared between MMR-Detect and MSI-Seq, we did find loss of function mutations in MMR genes. So we think this is a false negative of IHC. And then in this case, we had 2 cases that were just uniquely called to this other classifier, MSI-Seq. But in fact, they were not mismatch repair deficient tumors. They were actually polymerase epsilon mutant cases. They also tend to have a high indel phenotype, which is why this is these are false positives. So that's just to give you that first comparison. We then went to the next step, which was to test it on more samples, on over 2,000 colorectal cancers, 700 uterine cancers in the 100 kgp, and over 4,500 already published whole genome sequence cancers, primary and metastases in ICGC and in Hartwig. And just to give you the sort of high-level summary, there was very high concordance between MSI-Seq and MMR-Detect, but MMR-Detect did have higher sensitivity, especially for tumor types other than colorectal and uterine. Because remember, a lot of what we know about mismatch repair deficiency has come from colorectal cancers and uterine cancers, where the replicative rate is extremely high, but you're not going to have that kind of replicative rate in, say, the breast. So mismatch repair deficient tumors tend to have a slightly lower tumor mutational burden as a result. So some of these TMB assays will miss mismatch repair deficiency in other tumor types. MMR-Detect also has higher specificity. And I'm pleased to say we applied for a patent filing as of last week in a very agreeable IP discussion between Cambridge Enterprise, CRUK CRT, and Genomics England. So thank you very much for your expedited help there. So I kind of talked about mismatch repair detect. We've also got another algorithm called HR-Detect, which was actually developed before Genomics England. I just want to highlight it to you to sort of explain what the steps are once you've created a clinical algorithm. And we'll be creating more of these, but there are steps beyond creating a clinical algorithm in order to actually get clinical utility. Because all we've got right now is something that predicts biology. What we want to know is, does it predict outcome? Is it prognostic? Is it predictive? And so I'm going to use HR-Detect as an example of that process. So HR-Detect is an algorithm to predict BRCA-ness, and that's because tumors with BRCA1 and BRCA2 deficiencies are believed to be sensitive to PARP inhibitors, and about 1 to 5% of breast cancers are believed to come from women who have inherited BRCA1 and BRCA2 defects. So we trained our algorithm on germline mutation carriers, BRCA1 and BRCA2 mutation carriers. We used machine learning methods similar to MMR-Detect and created this classifier called HR-Detect. Okay, so HR-Detect high scores predicts BRCA-ness and low scores predicts not. And when we applied it on the 560 whole genome sequence breast cancers here, we found a surprisingly high proportion of cases actually had high scores, 22%, not 1 or 5%. That's 22%. That's 1 in 5 women could have a breast cancer that may have BRCA deficiency, which has implications for the options of treatments available to them. So if we look at them in more detail, we find that there were the 22 cases that we knew had germline mutations because we recruited them, but we uncovered another 33 families with BRCA1 and BRCA2 defects, so opened a can of worms there. We identified another 22 women who had somatic mutations in BRCA1 or BRCA2 or promoter hypermethylation of BRCA1. So we did have epigenetic data for some of these cases. But for a third of the cases, a third of the patients who had high HR-Detect scores, who had the signatures of BRCA-ness, we could not find a genetic or epigenetic driver. So if you do targeted sequencing, you are not going to see this 1/3 of cases who have the signatures, but they must turn off the pathway in some way that we don't fully understand yet. Okay, so at this point, we have an algorithm just like MMR-Detect, right? We've got something that predicts biology. Does it predict outcome? Okay, so then we collaborated with this team, UN staff in Åkerborg in Lund in Sweden. This is a whole genome sequencing study in the population and the whole of the south of Sweden. These are names of district hospitals, just like what you've done in Genomics England. These are just district hospitals in the south of Sweden, and every woman was recruited to the SCAN-B study who had breast cancer. And they've got thousands of tumors. We can't afford to sequence thousands of tumors, but we did sequence triple negative breast cancers because this is an area of unmet clinical need. We found that more than half the cases had HR deficient tumors. This is something we didn't expect. We know that triple negative breast cancers tend to have a high amount of BRCA deficiency. Okay, does it predict outcome? So the high HR-Detect scoring cases here are this black line in this Kaplan-Meier plot, which shows that if you have a high HR-Detect score, you're more likely to do better on standard of care in Sweden at that time. And this might mean that this is a cohort of patients that if you give PARP inhibitors or platinums, they'll actually do even better. But there's also this cohort that have done really badly. In fact, no better than the women who don't get any treatment at all. So we're really not doing these women any favors at all. Okay, the second point to make is that this cohort, we can only find the genetic or epigenetic driver in a proportion of them. So we take this group and we split them into the ones where we can find a genetic or epigenetic driver in black versus the ones that we can't, and we see no difference, meaning that the signatures on their own are capable of prognosticating outcome, right? So it doesn't matter that you can't find the genetic or epigenetic driver, the signatures can do the prediction. And that's important because, again, if you did targeted sequencing of BRCA1 and BRCA2 alone, or even PALB2 and RAD51C, you're just not going to detect this gray group here. And in fact, you're going to miss some of that black group as well, because some of these are due to promoter hypermethylation. Okay, so taking this HR detect approach, we can prognosticate. Let's go a step further. Patients with newly diagnosed treatment-naive triple-negative breast cancer were then treated with PARP inhibitors for 2 weeks prior to surgery in a window trial, proof of principle phase 2 trial called RIO. This was in collaboration with Nick Turner's lab in London. And again, more than half the cases had high HR detect scores. And again, just under a third, we cannot find a genetic or epigenetic driver, just like before. Right, but we also did some functional studies here. So we do find that there's a correlation between patients that have high HR detect scores and RAD51 foci formation. So there are functional HR deficits that correlate with high HR detect scores. Furthermore, we looked at circulating tumor DNA dynamics measured at baseline and at the end of treatment after 15 days. You can get an index, which is a measure of the number of copies at the end of the 15 days to the start. And basically, if you have activity to rupacarib, to the PARP inhibitor, then you'll get a very low score because the number of ctDNA copies drops, right? So you'll find here that the high HR detect scoring cases have got low CDR15 indices. So in this study, we show that HR detect high scores have a functional HR defect, and this correlates with activity to PARP inhibitors, rucaparib in triple-negative breast cancer, opening doors to exploring applications of this signature-based biomarker as a stratification tool in randomized controlled trials going forward. So we've demonstrated that we can use mutational signatures, apply machine learning methods, create algorithms, but there's a huge amount of work that goes into demonstrating clinical utility, which is to demonstrate prognostic potential and predictive potential as well. So a couple of other things to point out to you. I'm sure I'm preaching to the converted here about why whole genome sequencing is so valuable, beyond the algorithm, beyond the mutational signatures. What else do the whole genomes give you? So I think, it was critical that we can also spot the poor responders, or poor responders, even if you look at my bad spelling. We've always known that in triple-negative breast cancers, there are cases that just do so badly, but we've never been able to identify them. But now we can. We can spot the ones who are going to do badly and spot the ones that are going to do badly and the ones that are going to do well. So that's important. The second thing to say is that a lot of clinical trials stratify patients on single driver mutations. So let's look at driver mutations across the cohort. These are all the drivers. Let's focus on one of them, PIK3CA, famous one. So, we've known that PIK3CA is enriched in the poor responders, which is why you see a lot of them down here. But you can see PIK3CA mutations throughout. So if you conducted your clinical trial and you stratified patients just on these mutations, you're completely ignoring the rest of the tumor context, where there may have been an a priori that the patient was always going to do well on the standard of care arm. Okay, there's also a cohort with an even worse outcome, which is one in the middle here, which has other bits of biology in there, other signatures that are indicative of, say, replication stress. And these are important to identify because that may open doors to them being sensitive to other kinds of treatments like ATR inhibitors or check kinase inhibitors. And last but not least, in this cohort, there are mismatch repair deficient samples, something we don't detect in breast cancers because we don't look for them. But if you did a whole genome sequence, you would be able to find them all. You would spot the ones who are going to do well. You would spot the ones who are going to do badly. You would spot the ones who've got the replication stress and the mismatch repair deficiency, all with one assay. It shouldn't be thought of as a binary test for one mutation anymore. We need to start thinking about whole genomes in cancer as the whole portrait. Okay, so that was me just doing my usual preaching about why whole genome sequencing is valuable. I would like to now just in the last few minutes focus on a case just to drive home this message why it's important to read the whole genome, not just look at a driver or a signature. We need to read the whole lot. Okay, so this is work done by one of my clinical PhD students, Sophie Moman. So this is a patient who is actually known to the XP clinic. So he's got XPC, which is DNA repair defect. So he's got a recessive condition. He's inherited 2 abnormal alleles from his parents. And historically, these kids with abnormal XP genes would get a lot of UV damage because XP genes are important in nucleotide excision repair, which is to fix UV damage. So in the past, kids would have terrible lentigines, they'd have an increased risk of skin cancers, but these days they get fantastic care, they get meticulous photoprotection, they do very, very well. Now, unfortunately for this patient, he presented with this violaceous tumor in the middle aspect of his left eyebrow, and it was diagnosed as a very rare condition called an angiosarcoma, a cancer of the blood vessel lining. Okay, so he had wide localization, no radiotherapy, and he got Mets within a few months, and then the Mets spread aggressively. He just didn't respond to any of the standard treatments for angiosarcoma. And by July 2018, this man was dying. When we did a whole genome sequence of his angiosarcoma, we saw a heavily mutated cancer. We found the typical drivers of anything. It wasn't really an angiosarcoma based on these drivers, but his mutational profile was classic of UV damage. And it would be so easy at this point to go, oh, it's UV, it's like a malignant melanoma, treat it like a malignant melanoma. But in fact, we have to interpret the whole patient. So remember, he's got those XPC mutations and he can't fix UV. This is normal for him. What's really driving his disease is this signature, which may look like, really unimportant and small, but actually it's an indicator of a very particular signature called signature 10 caused by polymerase epsilon proofreading mutations. And in fact, he did have a driver, but it was in a minor subclone, so required manual curation in order to find it. It was missed by all the algorithms. So that was important because Pol E mutated cancers tend to be sensitive to checkpoint inhibitors, and immunohistochemistry in the Mets, but not in the primary, showed that he might be sensitive to PD-L1 checkpoint blockade. Indeed, here are his scans pre-treatment showing significant lesions in many places. And after only 3 cycles of pembrolizumab, everything is melting away. And as I understand it, his last scans were a few months ago, everything was clear. So we're 2 years in and he's done extremely well. And so that is a nice anecdotal story, but it really underscores the fact that we need to read the whole genome and take everything into account, the drivers, the signatures, the germline, and interpret it, not just pick out certain things. Okay, so in my last couple of minutes, I had intended to talk a lot about signatures in Genomics England, but we haven't published it, and I was a bit afraid that we would get scooped. So I'm just going to give you high-level insights. You'll have to invite me back another time. And this is work done by Andrea De Gasperi. So I'm going to give you really, really small high-level insights, but and it says here, do not post. Okay, so my team have performed holistic cancer genome profiling, like I described earlier there, on 15,838 of the cancers in Genomics England. This was version 8. We have done a lot of work on signatures on 12,222. That's because we're spoiled. We ignored all the FFPE samples and the lots of high PCR cycles. So we focus on flash frozen samples. And also we focus on 18 tumor types because those 18 tumor types are present in the ICGC and Hartwig cohorts that are out there. So over 6,500 whole genome sequence cancers that are in the public domain. That was critical because any new findings we made in Genomics England could be instantly validated with these external cohorts. So that was valuable. We have the 18 tumor types down here. We have all the different mutation classes, single, double subs, indels, rearrangements. Purple is gel, blue is ICGC, and green is Hartwig. You can see that the data is actually beautifully comparable. You can see many more Genomics England samples in many of these tumor types. And one of the things that we showed before was that mutational signatures show some tumor specificities. So the signatures are slightly different from one tissue to another. And indeed, we kind of looked at the mutational signatures of all the different tumor types, all these tumor types in Genomics England, in ICGC, and in Hartwig. We did the extractions independently, and then we just agnostically compared all the signatures that were derived between them. And you can see these beautiful diagonal set of deeper colors showing the proportion of matching signatures are much higher between the same tumor types, sort of reinforcing this sense that there are some tissue specificities in mutational signature. mutational signatures, which some of you may think is entirely academic, but actually matters when you are trying to assign signatures to your samples in order to do clinical stratification. When we compare our signatures here in blue to those reported in COSMIC, which is the standard sort of reference set, this is the new COSMIC, so there's quite a lot of them. We can validate 40 of them, which is what we are showing between here. And we also identify another 41 with high confidence. There's a whole bunch more with less confidence, or were only identified in ICGC and Hartwig, so we're not going to talk too much about them. And this just shows the sort of similarities between the 40 signatures here. So there's pretty good concordance here, but there was a lot of new stuff. And this is just single base substitutions, by the way. There's also some other stuff, double subs, indels, etc. Okay, so in the interest of time, there is plenty more to talk about on the academic content of mutational signatures. I don't want to go into it, but I do want to emphasize that it is important to use the signatures correctly. Here is a PCA plot of recurrent attempts to extract mutational signatures from a data set. And you'll see here that there are some signatures that are easy to get right, easy to extract, and therefore easy to assign to a sample. And there's a whole bunch of signatures that are difficult to get right, easy to get wrong, and could be assigned incorrectly. And I'm highlighting signature 3, which is our BRCA signature, because it gets called way too much in too many samples. And I caution this because I want signatures to be used well. So this is just a word of caution. But of course, that's why we create all those algorithms, in order to help people use signatures more effectively going forward. So I think I'd just like to end on this point, which is, how do we accelerate the process of converting mutational signature algorithms into clinical utility through perhaps an effective partnership with Genomics England? My team are kind of creating this Steiner cancer genome interpretation tool to help all of you use cancer genome data and to interpret cancer genome data going forward. You'll be able to have the visualizations and the thinking that we apply to cancer genomes, so that you can do that interpretation in your respective genomic medicine centers. So that's it from me. I'd like to thank an extensive list of collaborators noted at the bottom half of this slide. The Genomics England team that have been super responsive and positive throughout. My highly international team, from whom I draw a huge amount of inspiration here in gallery mode, and have been super throughout this really bizarre time with COVID. The people highlighted in red have done a particularly large amount of work presented today. And of course, I'd like to acknowledge the leadership of Genomics England, ICGC, and Hartwig and CPCT for access to the data. All the different funders, CRUK, NIHR, Joseph Steiner, welcome, and of course, patients, families, and clinicians that have contributed samples described today. Thank you very much. Happy to take any questions.
Thank you, Serena. So there's a couple of questions in the Q&A. Otherwise, can people put their hands up if they've got questions? So I'll just read out, there's an anonymous one. How did you feed back the new germline BRCA1 to mutations identified through HR Detect?
Okay, so we haven't done that yet for Genomics England. I guess that's something that we're going to have to do. But in the previous cohorts, we had to go back to the clinicians that provided the samples. And in some cases, because these were the earliest genomics assays, they had to adjust ethics, etc., to kind of feed back information, but it did happen. There was no pathway, but it's something that we do need to put in place for Genomics England going forward. Yeah.
And there's a sort of related, how does HR Detect compared with other stratification methods, including, I just jumped out of sight, including those based on non-DNA data?
So non-DNA data, like functional RAD51 foci assays, for example. So we had that in the Rio trial, where we did functional RAD51 foci assays. That was in Nick Turner's lab. There's very, very good correlation there. Yeah, but I think that was a small study. That was only 34 patients. So I think we'll need to do that in a greater cohort. I think there are people in Holland who are also doing something similar there.
And how many same questioner, how many patient cohorts have been used to test stratification methods, and how do these cohorts differ in terms of demographics, ethnicity, etc.?
So that's a great question. The 560 cohort was the first one that we trained the algorithm, that we tested the algorithm on. That was actually a mix of about 300 Western and about 150 or so Asian cancers, so from Korea and Singapore. So there were mixed ethnicities. HR Detect behaves the same way regardless of the ethnicity of the patient, and it's because it's a somatic signature. It's just a outcome of a DNA repair pathway defect. So we haven't seen any differences in the signatures itself. If there is a question about is the frequency any different? Now that one I think we haven't been able to answer yet because we haven't sequenced the same numbers of samples of Asian and Western tumors. Certainly in our cohort, there probably was a higher proportion of HR deficient tumors amongst the Asian population, but I think that's because of the nature of the samples that we got from Asia at that time. If there's more triple negative breast cancers, and you're going to see a higher proportion with high HR Detect scores.
And I mean, I'm not quite sure what this is directed to. Are the mRNA detect classifications available within GEL? I guess that probably means within the research environment for other researchers.
So yeah, we can make that available as soon as the paper is published. But please note that the MMR detect classification is only for some tumor types, not for all the tumor types.
Sure.
I will also say that we're already working on an improvement on MMR detect. So yes, there's new stuff coming.
Somebody is asking about you said mutational signature 3 is overcalled. How do you suggest that's avoided?
So that's a great question. And I think the way to do it is to use algorithms like HR Detect. So signature 3 on its own, it has a poor specificity because you can get signature 3 elevation for a lot of different recombinational defects, and presumably it's the result of various different translational polymerases. But if you want to try to understand which of your samples have got a HR deficiency, the thing to do is use HR Detect. So HR Detect, just critically, it uses signature 3, signature 8, rearrangement signatures 1, 3, and 5. I'm going to get this wrong now. My team are going to be laughing at me. Microhomology mediated deletions and copy number signatures. So it uses 6 signatures instead of 1. And when you use a signal that comes from 6, you have 6 different signals, your sensitivity and your specificity goes up. So signature 3 is still used, but in combination with other signatures.
I see there's somebody's put their hand up, Maria Kochinta. Maybe can they be turned on so they can ask a question? Yes, go for it, Maria. You're unmuted. I think you have to unmute yourself. Maria. Well, maybe she's trying to unmute herself. I think there's one more. Could you say more about why you think the UV component in that case you mentioned wasn't significant?
Because he is an XPC mutant, so he has, XPC not working at all, and XPC is necessary in nucleotide excision repair. I think he can't fix UV damage as well as you and I can. And so he is more likely to have the patterns of UV damage in his normal cells. So, I mean, we would all, if we could sequence all our skin cells, we would all carry UV damage as well. And in fact, a surprising amount, it's sort of tens of thousands of mutations can be seen in all our normal cells. We have another piece of work that we've put in bioRxiv where we've looked at iPS cells derived from skin, and it's, it's more than cancers, in fact. So I think a lot of UV damage is actually just consequential and just a circumstantial thing. But it was really striking in that case because that's the first thing you see. But the important thing there was that actually the thing that was the signature that was a bit less prominent was probably the thing that was more significant, more biologically significant, because polymerase epsilon is a critical polymerase in reducing the error rate in your cells.
Maria's still there, but she's not unmuted. So, well, okay. So I just wanted to ask one more thing myself. So you showed those stratifications, and you said that the poor performers were actually doing as badly as not getting treatment at all, which then suggests, of course, because all these treatments are detrimental generally, they're nasty things. Have you got a threshold where you could actually confidently say to clinicians, there's no point treating this patient, or is this like polygenic risk scores? We're just not there yet in terms of there's always going to be false positives in those groups, or ethnic sub-classifications where you haven't got the balance right?
Yeah, I think, we probably do need to do a few more clinical studies to just replicate what we've shown. But there's no question that we can identify the ones who are HR detect high versus HR detect low. So that I think is under no doubt. But
is it good enough for clinical use though?
Yeah, so this is it. I mean, you know, you saw the curves. Wouldn't you want to know? I mean, I would want to know what my tumor type, where my triple negative breast cancer sat. So I think this is the point where we do need to start doing the phase 3 trials, the randomized clinical trials, to try to understand that that's the next step. But to do that, of course, we need to get accreditation for the tools. And so that is the next very expensive step for me, which is to get accreditation for the tools to go into randomized controlled trials. Because if you're using an algorithm for stratification, you need to have accreditation, basically. But yeah, that's the next step. The next step is randomized controlled trials to answer exactly that question. Is it good enough now to use as a clinical test?
And of course, there's whole bits of NHSX, for example, have an AI team specifically, you know, in this, you know, how can we get more AI algorithms effectively into the NHS? So, all right. So I don't think we're going to get Maria, and I think that's the last question. Oh, there's one more. Do you see cases where patients with high HR detect scores do not respond well to inhibitors? I guess probably you do.
I'm sure we will. At the moment, the only one that's been done is that Rio trial, which is treatment-naive triple-negative breast cancer with PARP inhibition. Remember, a lot of PARP inhibitor therapy usually comes after rounds and rounds of platinum, and then they go into a PARP inhibitor trial, which I always think has enriched for patients who will have developed intrinsic resistance to anything that would go against the need for PARP. So I think we need to do more studies to answer that question. I do know that working on ovarian cancers, we do see cases that have got resistance to PARP. But as I said, that's after rounds and rounds of platinum already. So, you know, I just think we need to do the studies correctly, and yeah, we need to do more of them.
And finally, just one more, I think, before we end and switch over. Is the data for hematological malignancy specific for multiple myeloma? Can we use these databases for miRNA analysis?
So I have a colleague in MSKCC, Francesco Mara, who has done quite a lot of hematological malignancies, specifically multiple myeloma and HR detect. So there is data out there. Whether you can use that, whether he's got miRNA data to go with it, I have no idea. I think you'd have to look that one up. But yeah, certainly others have applied HR detect in other tumor types as well. Okay,
so thanks very much, Serena. Great talk.
Thank you very much.
And we'll now switch over to the second talk. And this is going to be a tag team between Alistair and Marcello. And they're going to talk about S3 loss of function causes a novel developmental disorder. So I think Alistair's going first, is that right?
Yep, that's right. So I'm just sharing my screen now. Do you want to just let me know if that's worked?
All good.
Great. Okay. Okay. So let me start the pointer as well. Yeah. So we're switching now from cancer genetics to rare disease now, and we're going to tell you a story about this gene called SCOBY3, and we found that loss of function variants in this gene can cause a novel developmental disorder. As Tim said, we're going to do this as a double act. So I'm going to start off talking about how we prioritize this gene using data from the 100K project, and then a few slides about some follow-up studies that we've done on the family that we identified. And then lastly, I'll talk about some of the other families that we identified through GeneMatcher. And then Marcello is going to take over and talk a bit more detailed about the sort of clinical comparison across all the families that were identified, some functional studies that they did, and also some mouse studies that have been done on this gene. And then if there's time at the end, some ideas for ongoing work that they're doing in Italy. So actually, this is a nice story to tell today because it was one of the first analyses I did upon getting access to all these genomes in the Genomics England research environment. And looking back at exactly what I did and aims, what I was trying to do. So firstly, I was trying to test out this script that had been provided by the GEL bioinformaticians. So this was called extract by consequence script. But at the same time, try and identify some interesting candidate variants. So as most of you know, a lot of the rare disease families in 100K GP, there are a lot of singletons, duos, and trios. I mean, there's a few larger families, but in general, strategies for finding novel genes like linkage aren't really going to work. So instead, we wanted to look across, instead of looking at just specific families, we wanted to look across all families. And also at the time, I was unaware that, I don't think there was any good global de novo call sets available. So we thought let's take another strategy. Let's just try and pick out some low-hanging fruits and just search for really rare homozygous variants and variants that seem to be likely to have a very severe consequence on protein function. So effectively trying to find individuals who are effectively gene knockouts. But so we didn't get too many candidates. We wanted to restrict this just to genes that seem to be intolerant to loss of function. And then also to make things work a little bit faster, just instead of searching through all 70,000 rare disease VCFs, just search through proband, so one per family, and families where there was likely to be some consanguinity. So I put together this sort of rough schematic diagram just showing you. So to get this script to work, we needed 3 input files. So what we define as a severe consequence, a set of genes that we think are intolerant of loss of function, and then I don't know if the point, and then these 286 file paths pointing the script to these VCF files, and then that generated a variant list. There was quite a lot of manual curation, even though we'd got rid of frame shifts. We knew those were going to be quite noisy. There's still a lot of curation, for example, stop gain in a exon that's quite often alternatively spliced out, for example, you might not think would be that interesting. But also there's still some artifacts that we had to remove. And then once we'd got this cleaner set of candidate variants, we then did a literature search, as I'll show, to identify what we thought were good candidate genes. And then I'll show some examples. We've done segregation and RNA studies, and then putting some genes into GeneMatcher. So this slide I'll just go over quickly, more for the bioinformaticians. So this was the extract by consequence script that was provided. So it was very simple. We just had to switch basically 3 bits of that script. We had to switch so that we input our, so first of all, this was the region file for 3,000 genes with this high PLI score from EXAC. The second input was just what we define as these loss of functions, so these SO terms. And then this last input was these file paths for these 286 Build 37 probands of Asian ethnicity. And then the output, we then did a additional filter for homozygous, so also had to be rare in the 1000 Genomes Project. And then this was just a very quick, crude way of removing artifacts. So this generated a candidate variant list of 9 very rare homozygous loss of function variants. So you can see some details about these here. It's quite a nice number to have. It's enough so that you think, oh, there's a good chance that 1 or 2 of them are going to turn out to be interesting, but it's not too many, so that you can actually spend some time reading about the biology of each of these candidates. So very quickly, this Scooby3 that we're talking about today, this patient had a very short stature and was annotated as a likely sort of skeletal, unexplained skeletal disorder. So there was a paper from 2004 that had done northern blot studies and showed very high expression in these osteoblasts, so these early sort of bone progenitor cells, and also very high expression in the femur and humerus bones. There was also a mouse model. Actually, there was a few mouse model studies in the literature. They were slightly conflicting. So one of them pointed out some skeletal anomalies. So all in all, it kind of made sense that the patient in gel had a skeletal condition. It seemed like a very good candidate gene to follow up. A second candidate gene was quite a well-known gene in terms of cell cycle biology, but it hadn't been linked at that time to monogenic disease, although there's now one family reported in the literature. So of these 9 candidates, actually 2 of them looked pretty interesting, and Scooby3 we're talking about today, but we're also collecting families for this other gene still. So this was the family that we came across in Genomics England. It was a family of Sri Lankan origin, but actually the information in the research environment, it coded the family as non-consanguineous, but we looked at the ROH regions, and the Scooby3 variant was in a large homozygous region, but also globally, right across the genome, I think about 10% of the genome was homozygous. And the clinician checked back, and actually, sure enough, the parents were first cousins. We then recruited the family to a local study, and we got some very nice images courtesy of Ataf and Melita at Guy's. So this photo is very nice. It shows this affected child next to his younger brother. So straight away you can see he's very short stature, slightly low set ears. He's got a slightly unusual sort of leaning forward posture, but also very pointy chin and triangular face. So quite an interesting set of features. We managed to get DNA samples for the 2 unaffected brothers, and neither of them were homozygous for this splice variant. But to be absolutely sure it was a loss of function variant, we wanted to get RNA and to see what effect it had on splicing. So this was nice because it was an experiment I didn't think was going to work. So it's always nice when it actually does. So Scooby3, as we already showed, it's very high expressed in bone, but in blood, it's very low. So GTEx, it shows it's less than 0.1 transcripts per million. And also the blood sample was delayed in shipment. So when I came to extract the RNA, we got sort of lowish RNA integrity score. So the first PCR didn't work very well, but then I did nested PCR, and we got a very nice Sanger trace showing basically it leads to 4-base extension of exon 17. So a 4-base extension, it then leads to a frame shift. So that confirmed that it definitely was a loss of function variant. Yeah, so that's so then when we'd done those sort of follow-up studies, we were confident enough about it to put it onto GeneMatcher. So at the same time, we put it onto the GEL research registry. This was now just under 2 years ago, and it turned out straight away we could see that there was already an established group working on this gene, and that was led in Rome by Professor Tartaglia. And actually already they'd started doing some functional studies. They'd already collected 3 families, which were all 3 larger than our family. So our family came in number 4. But by the time we wrote it up, there's actually, in the end, there's 9 families. Family 9 actually went into the manuscript at revision stage. So they're mostly a mix of sort of pretty severe missense, but a lot of stop gains and frame shifts. There was a second family with a spliced donor site variant, but here they couldn't get any biological samples. So instead they did a mini gene assay. And again, it led to aberrant splicing, although here it's slightly different. There's multiple alternative bands. So it's a sort of complex splice products being created. And then there was, as well as that, there was one slightly exotic variant in a Saudi family, which comprised a deletion of exon 8, but also an inverted duplication of the previous intron. And this was, yeah, homozygous in a Saudi patient. So it's a nice collection of variants in 9 families, I think in total 18 affected individuals. But if you look at all the different countries where these families came from, it shows you that we needed to sort of, it's such a rare disease, we needed to sort of extend the search globally, if you like, to collect all these families. So that's all I was going to say. So at this point, I'm going to carry on presenting, and Marcello is going to carry on from this point. Okay.
Thank you very much, Ali. Thank you even to organizers for inviting me into this great seminar. So as Alistair said, so we collect 9 families and 8 cube tree variants. And this collection was by networking powered by GeneMatcher. The clinical profile was quite consistent and characterized by, as is shown here, short stature, microcephaly, long and triangular face, dental anomalies, and a large amount of skeletal defect, including bone immaturity. So can I ask Alistair to change slide for me, please? Yeah. So next one, yeah, thank you. So in this talk, I want you to have very images about SCUBA3 and what SCUBA3 does. SCUBA3 is an extracellular actin protein. Structurally, is composed by an N-terminal AGF domain as the first part of the protein, and a C-terminal CUB domain, the last part of the protein. The first one mediates the interaction between proteins within the extracellular matrix. The CUB domain is required for binding receptor of growth factor or morphin proteins. So SCUBA3 is expressed mainly in osteoblasts and craniofacial regions, and as well as in limb buds. Basically, it works like a co-receptor or auxiliary protein. And in the next slide is provide how the auxiliary proteins are important in development and physiology, as pathogenic variants in genes encoding these proteins can dramatically affect development. So going the next one, please. So with the intent of clinically characterize the condition, we noticed that another disorder caused by mutation in BMP2 had the same, quite the same clinical picture on what we observe in outpatient. In the next slide, I will provide just in the left side of the slide a cartoon in which it's provide the BMP signal. A BMP signal consists in a ligand. There are 3-4 ligands in 2 kind of receptor, R1 and R2. And a large amount of co-receptor or auxiliary proteins. Inside the cells, this BMP signal has a system of transduction mediated by SMAD protein, in particular SMAD1, 5, and 8. This is definitely required. This is definitely required for the gene expression, the control of gene expression, which is important for morphogenesis and organogenesis processes during embryo development. So in the right side of the slide, there are the first experiment we have done. So we tried to understand which were the protein interactor of SCUBA3. And after immunoprecipitation assay, we demonstrated that SCUBA3 physically interacts with receptor ligands of BMP signal pathway. So since then, we were so happy because we were confident that we were in the right direction. So in the next slide, is the represent experiment in which we try to understand whether SCOBY3 could affect the BMP signaling. So in the first time, we knocked down SCOBY3 in the mouse mesenchymal progenitor cells, and we realized that after that, the downstream signal was definitely reduced. On the other side, when we overexpress SCOBY3, the same signal was so increased. So in the next, once we have understood that the physiology of SCOBY3, we try to understand the pathogenic mechanism of this protein when in turn. So to do that, we generate 3 representative mutants. One was truncating, one was missense affecting the Q domain, and one was missense affecting the AGF domain. While the truncating and Q domain mutants definitely impair the localization of SCOBY3 to membrane, the missense mutation involving the AGF-like domain affect the capability of SCOBY3 to homo or heterodimerizate. So they making a complex between other SCOBY proteins. In the next, we try to recapitulate and try to generate the disease model. And in the first cartoon, on the left, this is the scenario in which SCOBY3 works. So when SCOBY3 works, it is able to bind the ligand BMP2 and receptors. So the final effect is powering the BMP signal. In the other cartoon on the right, when SCOBY3 doesn't work, it's over there, the BMP signal is still active, but with low efficiency. So in the next slide, we try to understand the anatomical impact of this putative gene disease. So we generate the mouse model in collaboration with Professor Yang, University of Taiwan. SCOBY3 knockout mice were live and viable, but they were also reduced in length and weight compared to other littermates. In addition, they showed some clinical features, just like facial features, dental abnormalities, and vertebral defects, very similar to those we observe in our patients. In addition, histochemical analysis allowed to demonstrate that there was a reduction of length of all long bones we test. So this model overall recapitulated the human disorder. So in the next, we have some confirmation by additional histochemical analysis. This demonstrates that all class of cells within the growth plate were affected. In particular, chondrogenic and osteogenic lines were definitely affected in knockout mice. And further confirmation I can provide in the next slide. further confirmation we had by using immunofluorescent analysis, and in the right part of this slide, this is the first attempt to fix that condition, at least in vitro. So we inside the knockout cells, primary cultured chondrocyte, we try to insert an extra gene, SCUB3, by using a lentivirus vector. And then we observed that all the SMAP protein return in highly expression. So this is the first data that suggests us that something could be fixed in this condition. So coming back to the phenotype in the next slide. So definitely we characterize a novel developmental disorder occurring in a consanguineous parents, characterized by growth retardation, microcephaly, craniofacial dysmorphism, dental defects, skeletal abnormalities, and bone growth defect, and in some instances, even conductive hearing loss. This work has been published in the beginning of this year. So the last is for taking home messages. So SCUB3 is a novel auxiliary protein acting as BMP receptor. So disease causing variant definitely perturb BMP2 signaling, and then SCUB3 knockout mice recapitulate the human disorder. Finally, we may say that SCUB3 loss of function underlies a recognizable developmental disorder due to defective BMP signaling mediated skeletogenesis. And with Alistair, we like to thank all collaborators involved in this study, all patients and family enrolled, and all of you for the attendance. Thank you.
Thank you very much, both of you. It's a very nice study of identifying a gene and then going off and doing this functional investigation to understand the mechanism. So I was going to ask a question about, obviously, if you look at the publication and the data you presented, you've got lots of different mutations which are essentially providing a knockout across this large receptor. Having found those, in terms of the defects, presumably there's other mutations probably in this protein which are not as penetrant. If you've done surveys, you've got a whole genome, at least in the Genomics England data set, have you looked for less penetrant mutations in this gene?
We haven't yet, but that's a very good idea. But one very interesting thing is also that this gene comes up in GWAS studies on height.
Right, yeah.
So that's exactly what the point you're making. So it's already been shown that intronic variants that maybe modulate expression a tiny bit have a very small effect in height that you can only measure by taking huge case control or huge GWAS studies. Yeah. Exactly.
So this is where rare disease genetics can point you towards common disease, at a weaker level in terms of polygenic risk scores. Right. So there's a question: are you looking into BMP signal transduction modulators as potential therapies?
I can respond on this. We're going to try a new protocol, a new project. So we're going to try it anyway.
Any more? So if you can either put questions into the Q&A or you can put your hand up. I'm trying to chase for who's putting their hands up. I guess having got this family, this set of families, are you finding any new ones since you've done the publication, has this stimulated other people to come up with the same observation of clinical phenotypes?
We are testing just one family. The problem had the same facial profile, so we are going to test by Howling exome analysis. So we will see. It's an ongoing test.
Yeah, it'd be very interesting to test. Is like, we think it looks like a recognizable genetic disease, but the real test of that is if somebody thinks, oh look, my patient looks like that.
Exactly.
Yeah. And just Sanger sequences it. Just if they've got enough confidence to do that, then that really proves it. So that would be interesting.
But you've got to sequence. If you're going to do that, you're going to have to sequence quite a large gene region, actually, given all the mutations you found across that region. Yeah,
I can't remember. It's like 30. Do you remember how big? How many? It's about maybe 30 exons or something. So
exactly, yeah,
quite a big job.
Yeah,
you'd have to be pretty confident about
it. It may be just easier to do the whole genome. Okay, so I don't think there are any more questions. Oh yes, there is. Sorry. Right. Someone says, "Great talk. With such clear mechanism, any utility in looking for hets with similar phenotypes in the same related pathway?"
I didn't get the question. Could you repeat, please?
Well, Catherine Lawler, she can ask herself, actually. If we can unmute her if she wants to ask. Catherine, you can, yeah.
Hello. Oh, great. Thank you. Terrific talk. I just wondered, with such a clear mechanism that you've demonstrated and such a really precise way that you discovered those variants in Genomics England to start with, do you think there's any utility then in going back to some of the genes in some of those related genes and cofactors and looking for other potentially matching phenotypes or related phenotypes which may be associated with hets or HOMs in those other genes?
Yeah, so you're saying like the BMP2 and the SCIBI3 are in the same pathway and have a similar phenotype. So is it worth looking at other genes in that same pathway?
Yeah, I'd be interested in your view on that.
Yeah, that sounds a sensible idea. I don't know if Marcello, do you want to say anything?
Yeah, actually, it's a very nice question. This is the other project we are going to propose. The point is right. Probably this is a tiny part of the big scenario in which other protein could be involved. We demonstrate just BMP2 pathway, but we realized that probably other pathway could be involved as well. Thinking about TGF beta maybe could be involved as well. The point is that SCIBI3 is an auxiliary protein, so it could work in other pathway at the same way at the same time. So we have to look in for it. Thank you.
Thank you.
Okay, so any more questions, anybody? If not, then I'd like to thank the speakers, the two of you, and Serena before.
We have one more
presentations.
Question.
Oh, we have, I think it's...
Eamon.
Oh yeah,
no.
Sorry, it's just popped up, there you are. I think maybe is that a statement or a question? Variants in BMP2 have a well-recognized and similar phenotype. That's for information, I guess. Okay, so thanks very much, everybody, both talks. And just before we all go, I'll just repeat. Can I put up the slides for the intro? Sam and Zan?
Sorry, just setting it up.
So just to remind you all, next slide. we have these talks regularly now, monthly. It's the last Tuesday of every month. So the next one is on 27th of April. If you want to see what research is going on, you can go to the research portal at that address. And even if you're not a member of the GSIP community, or you can go and look and see the lay summaries of all the projects that are active going on inside the research environment. There's more than 400 of them, including all the clinical projects as well. There are lay summaries there too. And if you want to keep up to date with recent publications coming out of Genomics England, the Research Collaborative, there's that Twitter account. And if you want to hear more about our engagement with the clinical community, the science community, and patients and public, and issues to do with ethics, there's a set of podcasts hosted by our CEO, Chris Wigley, being released pretty much weekly now. So go and have a look at those. And if you have any questions, then there's GSIP help, which you can mail at Genomics England. And next slide. That's it, actually. Yeah, so if you've seen the benefit of using this data set, it's free access to this data. You just have to work inside our research environment and go through ethical approval in terms of joining a domain and getting your research institute to sign up if they haven't already. There's more than 400 research institutes around the world who've signed up to terms of use, and more than 3,000 academics who've applied for access. And we have several hundred, on a regular basis, who are accessing inside this research environment, carrying out analysis. So it's a large consortium effort now, looking at more than 100,000 whole genome sequences and a huge amount of associated clinical data, which is being added to every individual in the 100K has consented to all their clinical data, if we can get access to it, being put into this research environment. And so data is being added on a continuous basis with updates of data sets every few times a year. So if you're interested, have a look at the website, or again, you can contact the team through GSIP Help. Okay, so thanks very much, everyone. Thanks for some great talks, and see you next month. Bye.
Thank you. Bye.